Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing TIFR Biology question paper conducted in the year 2017. If you are new to my channel, please do like, share and subscribe. Then next one, that is coming to the year 2017. When antibiotic sensitive bacteria are spread on a plate containing antibiotic, colonies of resistant bacteria grow. The best explanation for this is the antibiotic is mutagenic. The antibiotic induces resistance through epigenetic effects. Cells only become resistant once they sense the antibiotic. Some cells in the original population were already resistant. That is more correct. They have asked for the best explanation. So D is more correct. Then next one. During growth and division of E. coli, the daughter strand is recognized due to. Now that is due to hemimethylation of newly synthesized DNA. Answer is option A. Then next one. Which of the following molecules is useful in protein phosphorylation studies? Now it is gamma P32 ATP that is useful in protein phosphorylation studies. Then next one. Place these steps in temporal order, first to last. Diffusion of neurotransmitter across the synaptic cleft, binding of neurotransmitter to its receptor, influx of calcium into presynaptic terminal, release of neurotransmitter, generation of action potential at postsynaptic membrane, influx of sodium ions. So the first one will be influx of calcium into presynaptic terminal. Then comes release of neurotransmitter. Then diffusion of neurotransmitter across synaptic cleft. Then binding of neurotransmitter to its receptor. Influx of sodium ions, and finally generation of action potential at the postsynaptic membrane. So the answer is 341265. Answer is option D. Which of the following statement is not a necessary consequence of meiosis? Chromosome number remain constant generation after generation. That is a consequence of meiosis. Each daughter cell receives the same number and kinds of chromosome. That is not correct. Not the same kinds of chromosomes. Each generation has a different genetic makeup than the previous one. That is also a consequence of meiosis. 4 daughter cells are produced at the end of the process. That is also a consequence of meiosis. So the answer here is option B. Each daughter cell receives the same number and kind of chromosomes. Then next one. Which of the following is not used for the generation of transgenic animals? It is next generation sequencing. Answer is option D. What is the correct order of action of these enzymes during replication? Helicase, primase and polymerase. So it is helicase, then primase, then polymerase. Answer is option A. Which of the following point mutations would be most likely to affect protein function? So TAA to TGA, they both are stop codons. TAA, TGA, TAG, they both are stop codons. CAA to TAA, TAA is a stop codon. So CAA is a coding for an amino acid. So the one which is coding for amino acid is getting mutated to a stop codon, means it is a point mutation and it will affect protein function. So B is the answer. AGG to AGA. Only the third base is being changed, or the third nucleotide is changed. So and we know that there is wobble hypothesis, and for the same amino acid as the third nucleotide is degenerate. Then CTT to CTC, here also same thing is happening. So these mutations will not be taken in, will not cause much effect on the protein functions. So the one which is causing most likely effect on protein function is CAA to TAA. Then next one, a mutation in an X chromosome gene, green, results in a green beard if a green bearded man marries a non-carrier woman. Now green bearded man XG and Y and marries a normal woman XX. Now XG is one gamete, Y is another gamete, and the female produce only one type of gamete, that is X. So G, X and X, Y will be the offsprings. So all daughters would be carriers and sons will not be having the green beard. Next one, bacteria isolated from hospital surfaces, that is furnitures or floors, are often very resistant to antibiotics. What feature of bacterial biology promotes this phenomenon? That is because bacteria can exchange genetic material. Answer is option D. Then next one, in resting skeletal muscle, contraction does not occur because. So what happens in muscle contraction is signal reaches the neuromuscular junction. The skeletal muscle excites and release of calcium from smooth endoplasmic reticulum. So calcium ions of cytosolic concentration increases and this results in muscle contraction. Now in resting muscle, contraction does not occur because there is very little calcium in the cytoplasm. Answer is option C. Then next one. In sucrose density gradient, what is the order of organelle sedimentation from lower to higher concentration of sucrose? Golgi, smooth endoplasmic reticulum, rough endoplasmic reticulum. Answer is option A itself. Then next one. We are studying a protein coding gene in an organism whose genome is not sequenced. The nucleotide sequence of 20 bases at the 5' end of its 900 base exon is known. What is the most efficient way to find the entire coding sequence of the gene? So what you have to do is you have to first reverse transcribe it, then PCR amplify the resulting DNA using a 5' primer against the known sequence and oligo DT primers, then do Sanger sequencing. Answer is option C. Then next one. In a purification experiment, a mixture of three proteins A, B, C are separated through gel exclusion or cation exchange chromatography. During elution, the buffer used is of pH 7. Which of the following statement is true? So here if you see, the isoelectric point is 11 for 45 kDa and pH is 7. So pH 7 is less than 11, pH is less than Pi. So it will be having a positive charge. Now 3, it will be having a positive charge. And 45 kDa, it is A. Is 45 kDa, it is smaller. So through gel elution, it will elute last in the gel elution. Similarly, A will be more positively charged and it will elute last in the cation exchanger. So the answer here will be A elute last in both the cases.